Hello, today we are going to learn about Oedogonium algae. It is a freshwater green algae. You can see these filamentous end-to-end connected cells of Oedogonium. This is Oedogonium. This is its reproductive structure called Oogonium. So we will see these things in detail. You can see that it is a chain-like uniseriate arrangement. Uniseriate arrangement means what? End-to-end connection. Okay, in a single line you can see this. Okay, so within that there is protoplasm which is having chloroplast chains, etc. We are going to see this. Okay, we can see this kind of algae in ponds, rivers, lakes, etc. Basically it favors the standing water habitat just like ponds, etc. So coming to its systematic position, whenever we are writing an answer in algae, if it is related to some genus, we have to write the systematic position first. So it belongs to the kingdom Plantae, division Chlorophyta, class Chlorophyceae, order Oedogonialis, family Oedogoniaceae, genus Oedogonium. Okay. Under this there will be multiple species. Actually, worldwide there are around 400 species of Oedogonium identified, and about 20 species of it belongs to India. So we'll see in detail about Oedogonium. First of all, coming to the ecology or habitat of Oedogonium. So it is found residing in stagnant freshwater bodies such as lakes, ponds, etc. And some species of it can grow in damp or moist terrestrial soils as well. For example, Oedogonium terrestris, Oedogonium rendave, etc. are found to grow in moist terrestrial soils. It can grow even in an alkaline environment where the pH is around 7.3 to 9.6, etc. So it can have a widely adapted type of life cycle. So it's okay, like it can live in favorable and unfavorable circumstances. We'll see how it is getting adapted to such unfavorable situations as well. Then there are freely floating species. There are species which are attached to the bottom of the water body. So different types of species are there. So this freely floating species forms a polyalgal patches or a mat-like structure at the top of the static water habitat. It is known as algal bloom. So a mat-like formation created by the Oedogonium creates algal bloom. Okay, in water bodies when these water bodies will be turning greenish color. These polygonal mats are created by secretions of different algal filaments. Just a value addition fact. Then there are different phases of growth. Okay, at first it will be in juvenile stage. Okay, after the spores are getting birth, it will be in the juvenile stage. That means in the baby stage. At that time, it is found in the benthic area or at the bottom of the aquatic body. Then when they are mature, they are coming to the surface and float on the surface to form polyalgal mats. As we have already told, it is created by the secretion of different algal filaments. Then Oedogonium filaments, just like we talked about wall box, we told that it will be having its favorable circumstances during the rainy season or spring and summer season. So similar is the case with Oedogonium as well. So this is the Oedogonium filament. You can see this is an end-to-end connected uniseriate arrangement, okay, of cells. So before coming to this figure, first of all come to this figure, because this apical cell is not demonstrated properly in this figure. So this is the apical cell. There are three categories of cells in Oedogonium. First one apical cell, second one basal cell, okay, which is also called holdfast cell because it is having these finger-like projections to hold onto some rocky bottom of the ocean or rocky bottom of the freshwater body, or it can also grow on the surface of other plants. Okay, so it is also known as epiphytic. Okay, so for that this holdfast cell is being used. So you can mention about that. So apart from the apical cell at the top and basal cell at the bottom, the remaining cells which are coming in between, okay, you can see the intermediary cells. These are called intercalary cells. Okay, these intercalary cells are the cells which will be taking part in reproduction, which we will see later. So coming to an individual cell of Oedogonium, this is an individual cell. It is having a proper nucleus at the center region. Then it is having a chain of chloroplasts. These strands are the chloroplast filaments which will be responsible for photosynthesis, and whatever be the output of photosynthesis, the carbohydrates or starch, that will be stored in the pyrenoid bodies. You can see the black dots in between, in the chloroplast strands, which are found at the junctions of these chloroplast strands. These are called pyrenoid bodies. Okay. So apart from that, there is actually a center vacuole in the Oedogonium filament. Actually, this is a cylindrical structure. Imagine a powder tin, okay, and a longitudinal powder tin, okay, which will be having hollow inside, okay, right. So since it is a two-dimensional figure and a longitudinal section which we have cut longitudinally, they have not represented the vacuole, but the central area, the hollow area, which is having a vacuole as well, which will be filled with some secretions and excretions, excreted materials of the Oedogonium. So that will be also there. So this is about Oedogonium. Now when we are studying about the reproduction phase, you will be mentioning about this Oedogonial cell and antheridial cell, etc. But before that, you can see in normal phase, this is the structure. Okay, this is the vegetative structure of Oedogonial filament without having any separate bodies for reproduction like Oedogonium or antheridium, etc. This is a vegetative filament, but it will undergo some transformation during the reproductive phase. Okay, so it will be forming an Oedogonium, antheridium, etc., which we will see later. So this is it. Now coming to a different thing. Oh yeah, one more thing. Actually, when we talked about this cell, okay, this cell or this cell, the same thing only. It is having a proper cell wall, a thick cell wall for protection, and it is actually multi-layered. The outer layer will be of chitin, the middle layer will be of pectin, and the inner layer will be of cellulose. Okay, this black line I am telling, okay, it is having 3 layers. Okay, outermost layer is chitin, second layer is pectin, and the innermost layer is cellulose. Okay, so this is it. Now this is the vegetative filament, apical cell. These are the intercalary cells, and this is the holdfast or basal cell. Now coming to this triangle shaped cap, which are found in some filaments. Not all filaments you are seeing this cap, but some filaments you are seeing this cap. This cap is an indication of the reproductive ability of this cell. This is one cell, this is another cell. Those cells which are having cap indicates that this cell will be going to do the reproductive function. Okay, so only those cells which are having this cap like structure, which will be having the reproductive ability, which we will see in the upcoming slides. Now Oedogonium cell structure, we have already discussed all of these elements. You can read it. Just for your note making, I have given this thing. Fine. Then thallus structure, we have already discussed about the apical caps and cell wall, pyrenoid, everything we have discussed. You can see this, read and write this. Then coming to the two species or two broad categories of Oedogonium, because it is performing, these two species are performing two different reproductive behavior. That's why we are classifying it into two different species broadly. One is Macrantha species and the second one will be Nenandra species. Okay, so in the reproductive slide we'll discuss about the behavior, exact behavior of Macrantha species and Nenandra species, but since this is an intro class, we just need to know what is this Macranthus and Nenandras. So in Macrantha species, see the point, mouse pointer, you can see that. Imagine this is the Macrantha species. This is a normal vegetative filament. Now during the time of reproduction, one of the cells of this filament will modify into antheridia. Okay, antheridia. You can see the antheridium. This is one antheridium. This is one antheridium, which will be having antherozoids. Okay, so in the normal filament only, one of the cells will be modifying into antheridium. This is the case of like Macrantha species. And in the Nenandra species, this is the Nenandra species. The antheridia is not formed in the normal filament, but you can see a tail-like small tiny filament. You see this? So this will be bearing antheridia at its top. So this is the only difference between Macarandus and Anandrous species. The Macarandus species are having normal vegetative filament, and one of the cells of this filament will be dividing into form Antheridia, but in Anandrous species it will be having a tail-like micro structure called this male filament, which will be bearing the Antheridia. This is the main difference between Macarandus and Anandrous species. And one more thing, these Macarandrous species can be actually monoecious or dioecious. Monoecious means what? Both Antheridium and Oedogonium, this is the Oedogonium, can be found in the same species, same filament. Okay, so it can be either in that way or it can be dioecious. It can be like this. In this figure I have represented the dioecious species because from this filament it is forming, one of the cell is forming Antheridia, and similarly there will be another thallus like this. So this is another thallus, it is bearing the Oedogonium. Okay, so I have represented dioecious species here. So this is in the case of like Macarandrous species, but in non-androus you can see always dioecious behavior, because we will see how the dioecy operates in non-androus species. It is like this. If I am briefly stating, this is the algal filament or Oedogonial filament. From this, one of the cell will be modifying into androsporangia. Okay, androsporangia, and from this androspore will be released and it will be going to meet the Oedogonium at a different thalli. Oedogonium is inbuilt in a different thalli and it will be attaching to a nearby cell of this Oedogonium and it will be forming a new male filament which will be bearing the like Antheridia at the top. So this is the case of non-androus species. So for it to operate, one of the thallus should produce androspore and another thallus should produce Oedogonium. So this is the case of non-androus species. Now about reproduction, I have just briefly mentioned reproduction consists of vegetative mode, sexual mode and asexual mode, but in this session we will be seeing only vegetative mode. Sexual and asexual we will see in a different lecture. So vegetative reproduction is simple. It can happen through two ways. One is fragmentation and then is echinus formation. Fragmentation means what? From the capsule area. Okay, from the capsule, imagine, sir, this is a capsule. From this cap area junction, one particular fragments, particular cell gets fragmented and it will be split apart from the main filament body, and that fragment will be forming a new thallus. Okay, and one more thing I missed to mention, if a particular cell is having one capsule that indicate one cap, that indicates that it has divided only once. If it is having two cap, it indicates that it has divided twice. So if it is three cap, it indicates that it has divided thrice. So the number of cap indicates the number of times the cell has divided. So that is one thing you have to write. Then echinid formation. So these are the echinids. What are echinids? When the proper water supply or nutrient supply is not available, or if it is a drought-like condition where they cannot release the zoospores which can swim and reach the Oedogonium, what it will be doing is, it won't be like making any zoospores or something like that, but it will do a modification in the filament. This is a filamentous body and you can see the chain of echinids. What are echinids? Echinids are formed by shrinking the protoplasm of the normal filament. You can see it is concentrated towards the center. This is the thick walled echinids, which is the shrunken protoplasm of the normal cell. Okay, so this protoplasm will shrink and it will be covered by a thick walled covering. This thick wall will be having proper food materials to overcome the unfavorable situation. So it is found in chains, and like if you summarize about acinids, you can say that acinids are large, non-motile, thick-walled spores. Okay, and their wall is attached to the parent cell. So this is the wall of the parent cell. It is attached. Its wall is attached to the wall of the parent cell. When the favorable conditions arise, they start to germinate. Okay, it will burst open and it will be forming new filaments when the favorable conditions are restored. So this is all about today's session. So we'll see the sexual and asexual reproduction about Macarandus and Anandrous in the upcoming session. Thank you so much.